Professor Montana. The title of his talk is Mirage, Splice-Aware Multiple Sequence Alignment of Protein Isoforms. Please.
All right. Thank you all for being here. So today I'll be talking about Mirage. This is a new tool for splice-aware multiple sequence alignment of protein isoforms. So as we all know from basic biology, eukaryotes encode proteins using exons that are separated by intervening introns that end up being spliced out. And one of the benefits of this method of encoding is that it enables cells to dynamically adjust the particular functions of their proteins through the use of alternative splicing, such as through the use of cassette exons. And so when we have proteins within the same gene family that are alternatively spliced, we refer to the particular splicing products as isoforms. So what we have are multiple proteins that are being encoded by subsets of a shared exon pool. And we're interested in producing multiple sequence alignments of these protein sequences because this will help us identify amino acid resolution homologies between these sequences that have valuable applications in a variety of bioinformatics applications, such as annotating post-translational modifications, analyzing mass spec data, and refining genome annotations. Of course, these isoform multiple sequence alignments are only valuable insofar as they're accurate. And when we're talking about an accurate multiple sequence alignment of isoforms, what we're talking about is a multiple sequence alignment where entire exons are either aligned when they're homologous or else not aligned. And the problem we encounter when using traditional tools for this problem is that traditional tools will identify spurious matches between alternative exons and then construct alignments based on those spurious matches that involve short runs of gaps as well as mismatches. For example, we can look at this collection of sorbas2 isoforms where the highlighted exon has been misaligned by clustal omega in 4 different ways. So, our new tool, Mirage, was developed with the specific goal of producing isoform multiple sequence alignments that respect exon boundaries. And the way that Mirage works is that for a collection of input protein sequences from the same gene family and species, we individually map each isoform back to its encoding exons on the genome. Then, once each exon in this set has been mapped back to its each sequence in this set has been mapped back to its exons, we proceed through the genome mapping coordinates and use those to produce our multiple sequence alignment. We refer to this process as transitive multiple sequence alignment because instead of aligning the isoforms to one another directly, instead we're aligning them through the intermediate of their genome mapping coordinates. And we can see that this gives us the desired result, that we have a clearly exon delineated multiple sequence alignment. And moreover, we're able to explicitly record the positions of splice junctions in these intraspecies multiple sequence alignments. So to give a particular example where Mirage has improved a multiple sequence alignment, we can look at these human DLG1 isoforms. We can see the 2 highlighted anchor motifs come from different exons and shouldn't be aligned to anything else. Mirage gives us our desired result, whereas these leading multiple sequence alignment tools all struggle to make sense of these alternative exons. To give summary statistics of Mirage's performance, we can consider the fact that within species we expect percent column identity of isoforms to be near 100%. This is because a collection of isoforms within the same species is being encoded by exactly the same genomic sequence, so we should see no variation. So we ran a test where we aligned a collection of over 80,000 protein sequences from UniProt that represented nearly 22,000 gene families. And we see within the 3 most represented species in this group that Mirage significantly outperforms its competitors. Now, once we're aligning between species, we don't have the advantage of being able to map each isoform to a common genome. For that reason, Mirage has to use a more traditional profile-to-profile alignment method for interspecies alignments, but Mirage has 2 unique advantages. The first is that it has fewer intraspecies multiple sequence alignment errors to compound in its downstream alignment steps. The second advantage is that Mirage is able to leverage its splice junction information into its heuristic scoring schemes. To give an example, we can consider this toy case where this leucine can be ambiguously aligned to either a valine or an isoleucine. Now, a traditional method will refer to a score, and based on whatever the higher alignment column or higher scoring alignment column is, it'll select that alignment. However, Mirage has access to information about splice junctions, and so it'll penalize splice junction misalignments and reward alignments that preserve biological consistency with splice junctions. So returning to our UniProt KB dataset, we see that Mirage significantly improves interspecies multiple sequence alignments of isoforms. Moreover, Mirage is competitive in terms of its runtime with its competitor tools, and these numbers scale with both dataset size and the number of CPUs available. An exciting byproduct of Mirage is that it's been proven to be a powerful tool for detecting dual coding exons. These are exons that overlap on the genome but are frame shifted off of one another by one or two nucleotides so that their open reading frames produce distinct peptides. And I've included an example from 2 human ACDY4 isoforms. Now, Mirage is capable of recognizing dual coding exons because it maps isoforms back to the genome. So anywhere where our mapping coordinates are suddenly offset by 1 or 2 nucleotides, Mirage easily detects that there's a putative dual coding exon. And now this isn't something new that Mirage has discovered, and there have been plenty of sort of pilot studies into the nature of dual coding exons. But previous estimates have postulated that there are about 1 to 200 genes with dual coding exons. Mirage, however, has noted that there are about 2,800 gene families that have dual coding exons in the UniProt KB dataset. Now, at first we were worried that these might just be artifacts of noisy splicing and might not have any real biological importance. To examine this, we've performed a series of preliminary tests, and here are some exciting results from one of those. So in this graph, we have on the x-axis the lengths of exons, and on the y-axis we have the percent of mouse exons with multiple open reading frames. So every point on this graph represents a mouse exon with a dual code that's a dual coding exon. Now the red dots are exons that have some human homolog. Maybe that human homolog has multiple open reading frames, maybe it doesn't. And we see, as we'd expect, that the percent of mouse exons with multiple open reading frames decreases with sequence length because it becomes more and more likely as you increase the length of the sequence that the 2 alternative reading frames will have a random stop codon in them. However, we can look at the blue dots, which are mouse exons with multiple open reading frames that have homologs to human exons with multiple open reading frames. And we see that in a significant number of these, the dual coding exons are preserved in mouse. Or rather, the dual coding exons that we see in human are also preserved in mouse. So this strongly suggests that there's some evolutionary reason why these dual coding exons are being kept around. All right. So with that, I would like to thank my wonderful collaborators at both the University of Montana and Cell Signaling Technologies. And I'll summarize by saying that traditional multiple sequence alignment tools struggle when aligning isoforms, but Mirage is able to improve the quality of isoform multiple sequence alignments and has been shown to be a powerful tool for detecting dual coding genes. If any of you want to talk to me at the poster session, I'd be excited to do so. And if you've got nothing better to do on this beautiful day, feel free to read the preprint of our forthcoming paper. I'll take your questions.
Hi, good talk. So I saw with for isoforms of the same gene that your tool performed better compared to other cluster O mapped muscle. What if you just took the exon, though, and just checked, like, would Mirage still perform better than cluster low, mapped, and muscle?
Yeah, if we were only looking at the alternative exons, these numbers would actually blow up. We would see that cluster, they'd essentially, if I can get back in time, they'd essentially just be doing exactly this behavior that we see in the misalignment, whereas Mirage would give us the perfect alignment of the alternative exons. Does that answer your question?
Yeah, thank you.
Great. Yeah.
Hello. Thanks, very nice talk. I had more a general question. Very often you have a problem that you have a lot of transcript isoforms which are annotated in human or mouse, for example. And when you go a little far in the phylogenetic tree, you don't get anything. So would it be possible with Mirage to rescue a lot of conserved isoform? Maybe you tried already, I don't know.
Let's see. I don't know the answer to that offhand. That's a good question. We could discuss that later.